important topic, that is plasmid DNA. A plasmid DNA is going to be a DNA present in the bacteria. It is not a normal DNA to be present in a bacteria. It is an extra chromosomal DNA. That is, apart from that DNA of a bacteria, another DNA arranged as a circular chromosome is known as plasmid DNA. So in this video we are going to see about the plasmid DNA. So a plasmid DNA is going to be present in the bacterial cells. These bacterial DNA, apart from the bacterial DNA, a plasmid DNA is going to be present. What is a gene? It's going to be taken place is, it's going to have a gene of origin, a gene of origin for the replication of this plasmid DNA, because this plasmid DNA is not going to replicate along with the normal bacterial DNA. It's going to replicate autonomously as we present as an extra chromosomal. So it is going to undergo the replication from the origin site, ORI site, and lead to the undergo replication. So a gene going to be present in the plasmid DNA is going to be origin for the replication, a replication site origin. The genes are going to be present in the plasmid DNA. Another important DNA to be present, which is useful for the bacteria as it was present in the bacterial cells, are known as antibiotic resistant genes. These antibiotic resistant genes are very important for this bacterial DNA, for this bacteria, because these antibiotic resistant genes creates the proteins that is going to resist the antibiotics, so that this bacteria can survive even in the antibiotics. So it increases the survival rate. So the antibiotic resistant gene is going to be present in the plasmid DNA. Here we said that plasmid is a small DNA that is going to be circular in shape, that is going to be present within a cell. Within a bacterial cell it's going to be present, but it was not along with the bacterial DNA. It is going to be separated from the chromosomal DNA, as plasmid is going to be circular, but it is extra chromosomal, that is not with the chromosomal DNA. It going to be present outside the chromosomal DNA. Therefore, it can replicate independently. It is going to be extra chromosomal unit and autonomously. As we said, it's going to replicate independently and autonomously. But this plasmid DNA is going to be naked DNA, which can be seen easily. It is not going to be encapsulated with proteins, but in general, a bacterial DNA is going to be encapsulated. For example, TMV virus. In general, the bacterial DNA is going to be present inside the capsid protein, capsid coat. But in the case of plasmid DNA, this plasmid DNA is going to be extra chromosomal, not inside the bacterial DNA. So it was not found to be inside the capsid coat. It was outside the capsid coat. So it is not encapsulated in the protein coat. Next, the number of DNA. A number of plasmid DNA can be varies. This plasmid DNA number varies is known as copy number, designated as copy number. So, one plasmid in a bacteria, or can be 1,000 plasmid, maybe in some of the bacterial cells. So, next, the conformation of the plasmid. A plasmid conformation changes based on their region, based on their situation. So, the plasmid DNA can appear as an equipotent circular DNA. In some cases, during replication, it can known to be found as nick open circular DNA. At the end of the replication during catenation formation, nick opening is very important. A relaxed circular DNA, relaxed circular DNA, plasmid DNA is going to be a normal plasmid DNA to be present. A linear DNA, a linear replication, and supercoiled DNAs taken place during the catenance formation. A topoisomerase causes the circular plasmid DNA to undergo supercoiling for the release of another daughter cell. So the supercoiled DNA is also a conformation. Most of this conformation to be present during the replication. In normal, it was found to be a relaxed circular DNA, a supercoiled or denatured DNA, which means during the denaturation, the plasmid DNA can be present as a supercoiled denatured DNA. These are the conformation of the plasmid. This is a normal plasmid structure. Example, PBr322 plasmid is used here. It is one of the plasmid to be present in the E. coli. So here we can see there is only a 3 sites. One is origin. A origin region is region where the replication is going to be started off. Replication start at this origin region. Next, it's going to be tet means tetracycline. Tetracycline is going to be an antibiotic. So a tetracycline resistant gene is the TET gene. In PBr322, tetracycline resistant gene is going to be present. That means the bacteria, E. coli bacteria, which is going to have this PBr322 plasmid, can resist the tetracycline, which means it can survive. A bacteria which has this PBr322 can survive in the tetracycline containing antibiotics too. Based on this only, for culturing, based on this only, antibiotics can be suggested. Based on the bacteria which causes the infection, its plasmid is going to consist of certain antibiotic resistant gene. Based on that, the antibiotics is suggested. So a tetracycline resistant gene is going to be present in the PBr322. So this plasmid presenting bacteria can survive even the tetracycline antibiotics can be given to the patient. So this is AMP means ampicillin resistant gene. So it can survive in the ampicillin antibiotics too. So the another antibiotic can be suggested when the infection affects with this bacteria having this plasmid. So it is very important in the medical terms. Next, a tetracycline, ampicillin, or the antibiotic resistant. As we said that one is a replication start type to be present for the plasmid, another is going to be an antibiotic resistant gene that can be a ampicillin or tetracycline. So these are the genes to be present in the plasmid, which is helpful for the bacteria, which is antibiotic resistant gene, which makes it to survive even in the antibiotics. Next, these N-del or HIN or E-core, BAM or SAL, these are all the endonuclease cutting site. So if another gene is going to be introduced into this plasmid, because a plasmid is going to be a vehicle, molecular vehicle, so it can carry another gene, which means a site of E-core means endonuclease can cut this site, which can causes a integration of another gene of interest, which this process is known as recombination. A gene of interest can integrated into the site where E-core 1 cut and replaced by this gene of interest, and causes it to introduce to another organism, and then synthesis a gene of interest producing protein or gene of interest product, so that it can be used in molecular techniques. It is involved in recombination techniques. So these are the cutting site of the plasmid. In general, the useful region for a bacteria is replication start site and antibiotic resistant gene. Next, we are going to see about the structure of a plasmid, as plasmids are found to be small circular double-stranded DNA. It is going to be present as 1 to 200 kilo base pairs in general. That means it can be present to about 1000 to 2 lakh base pairs to be present in the plasmid. Next, we are going to see about the important region, that is a gene which is helpful in the survival of the bacteria is going to be antibiotic resistant gene. That can be tetracycline, ampicillin, or etc. They can also carry the genes for autonomous replication, that is the origin site where the replication is going to be taken place at the